Afternoon. I apologize for the technical delays. However, I'm welcoming you back to the second collision and the globe track, which is focusing on medical technology sector. To moderate this session, it gives me a great pleasure to introduce Manuela Cirilli. Manuela joined CERN Knowledge Transfer Group in 2010 after having studied both physics and science communication. With the KG Group, Manuela brings her unique blend of competences in the technical domain, medical applications, and communications. She is a deputy group leader since 2017 and currently leads the medical application strategy support team and the communication and training section. Thank you, Manuela.
Thank you so much, Anelia. I'm delighted to introduce our speaker for this session now, Dr. Martin McKeown. Martin is the director of International Center of Excellence for Parkinson's Disease. This is the Pacific Parkinson's Research Center at the University of British Columbia in Vancouver, Canada. And he's also professor of medicine and adjunct professor in electrical and computing engineering at the same university. So Martin is a clinician scientist and he sees patients with mostly Parkinson's disease, and he also conducts research to help people with Parkinson's. He came to CERN during his undergraduate work in engineering physics, so it's a very curious trajectory, and we are all very curious to hear about his exciting research and also how training at CERN has been helpful to shape his career. So the floor is yours, Martin.
Okay, thank you. Unfortunately, I do have a few technical difficulties. I can't see any of the slides. I just see my own reflection.
We still have technical difficulties.
Okay, thank you. I'm not sure I can advance the slides. So can someone advance the slides for me, or? Okay, thank you. I will just assume someone can advance the slides for me. So thank you very much. I'd like to thank the coordinators of this event for inviting me. It's a great pleasure to come and talk to people. As Manuela mentioned, I'm a neurologist, a physician who deals with people with brain diseases. I mostly see people with Parkinson's disease, and we're involved in a number of projects. But I thought I would focus on two where I think my prior physics background helped me in these particular areas. And those are looking at novel treatments for Parkinson's and also disease monitoring. So if we could go to the next slide, please. So what is Parkinson's disease? Well, it's a neurodegenerative condition. And what this means is that there are certain cells in the brain that die prematurely. And this is probably, it's poorly understood, but it's probably a complex interplay between genetic and environmental factors. And the most prominent cells that die in the brain prematurely in Parkinson's are cells that secrete the brain chemical dopamine. So many of our treatments are related to replacing this missing chemical. Parkinson's is the second most common neurodegenerative disorder after Alzheimer's disease, and it affects up to 2% of people over age 65, and if you get into your 80s, it can be as high as 4%. It affects all races, and so it really is a worldwide phenomena. It seems to be increasing in prevalence, although we don't know whether or not we're just recognizing the symptoms more. But it certainly seems to be more prominent now than it was several years ago. Next slide, please. So about 50 years ago now, as I mentioned, because one of the most prominent neuropathological changes are the loss of these cells that secrete dopamine, it was found that replacing this brain chemical dopamine through medication was an extremely effective therapy. And so about 50 years ago was the introduction of a medication called levodopa, which is still the primary medicine that we use to treat this disorder. That was expected. What was unexpected was about 30 years ago when it was discovered that not only treating abnormal brain chemistry, but treating abnormal brain oscillations is also a very effective and proven therapy. And fortunately, who knows a lot about oscillations? Well, physicists are very good about this. And so this is why I think the physics community has a role to play in looking at these new therapies. So next slide, please. So this gives you some idea of whether or not we are asymptotically approaching the limit of what we can effectively do with medications. So the pharmaceutical industry, as you know, is a multi-billion euro industry, and they have very strong financial incentives to develop new medications. I just looked at Alzheimer's because it's even more prevalent than Parkinson's, and so there's even more financial incentive to develop pharmacotherapy. And you could see here that recently they tried to put through a drug, and it would have been the first drug in 20 years to slow the progression of cognitive impairment in Alzheimer's, and it was unsuccessful. And as I mentioned, for Parkinson's, we're still using the same medication that was introduced half a century ago. So this tells us that we need to be looking at alternate approaches for therapy. So next slide, please. So this has prompted people to look at techniques of brain stimulation. So on the left there is so-called deep brain stimulation, which is discovered about 30 years ago, and it is now used worldwide as an effective treatment. However, it's open brain surgery. It requires you to drill holes in the skull, insert electrodes deep inside the brain, and then have an implantable stimulator and a battery pack placed under the skin that sometimes has to be replaced maybe every 5 years or so. So this obviously, anytime you insert a foreign body into the brain, there's risks associated with bleeding and infection and so on. So there's a lot of interest in non-invasive stimulation, which you can see on the right-hand side of the screen. And it is possible to activate the brain by either applying focal ultrasound, applying powerful magnetic fields in transcranial magnetic stimulation, or applying electrical stimulation, either with a complicated waveform or stimulating behind the ear, which stimulates the vestibular system or the inner ear. Our lab has focused on the left side, which are these potentially portable areas because they require small stimulators that people could carry around, for example. So if we could go to the next slide, please. So our lab has focused on stimulating the vestibular system. So the vestibular system is a very curious system. It's a very old system that's seen in very low animals, and it's related to knowing where your brain and the rest of your body is in space. And it seems to influence very wide areas of the brain. And it's used a lot for visual stability. So when you're walking, the reason why the video frame doesn't appear jerky when you're walking is because of the vestibular system. It's important for balance. It's important for knowing where you are in space. And it's important for controlling things like the blood pressure. So, for example, when you stand up, the blood rushes to your legs, and so there has to be a reflex that maintains the blood pressure so you don't feel dizzy and enough blood gets back to your brain. And how does the body know that you've stood up? Well, it's the vestibular system. And so it seems to be a very ubiquitous system that's used for many different things. A few years ago now, one of the things that we discovered in our lab was that if we stimulate the system electrically by just putting electrodes behind the ear and give a very low stimulus, less than a milliamp, uh, and at a level so low that people can't even feel it, that we were able to manipulate brain rhythms, and including the brain rhythms that seem to be abnormal in Parkinson's. So this excited us because it seemed to be a non-invasive way where people don't even feel it, where we can manipulate the brain oscillations. And so we think that this would be a potentially complementary therapy to medication. We don't think it will replace it, but we do think that there are certain aspects where the medicine is not working sufficiently well, where we may want to complement it with this neuromodulation therapy. So next slide, please. So this is recent work from my former PhD student, Sujin Lee. And what she did is she looked at different types of waveform stimuli. So as I said, physicists are very good at oscillations and looking at different waveforms that we could use. So what she did is we looked at a simple motor task where we measured reaction time. And what we did is we started off with a standard stimulus, which is a noisy or stochastic stimulus. And this is what is being used by our lab and other labs around the world as a potential treatment for Parkinson's. And we used that as a baseline. And what we did is we tried many other different stimuli. There were multi-sign stimuli. There was a simple linear superposition of sinusoidal waveforms in across a finite bandwidth. And what we did is we looked at the effects of improvements in reaction time compared to the baseline, which is the stochastic stimulus that's normally used. And what she found is that many of these other band-limited multi-sign stimuli were better as far as improving reaction time. But the key point was the optimum stimulus was a function of the individual. And we think that part of the reason why these technologies have not been more successful is they seem, if you use a one-size-fits-all stimulus, it works somewhat well for everyone. But what you need to do is choose the optimum stimuli for that individual, which has the capacities to significantly improve the benefits. And in medicine, we call this precision medicine, which is kind of an odd term, but it really means customized. So what we want to do is customize the stimulus to the individual. And this is why we are getting into data-driven approaches for this. So next slide, please. So this is one approach that we're doing. Now, if we want to test what is the best stimulus to, say, improve reaction time, obviously it's impractical. We have an infinite parameter space. We can't just have someone try millions and millions of trials of a motor task to check reaction time and check every different stimulus. So we have to be a little more clever about that. So one of the things that we're doing is we're using some brain monitoring technologies, including the electroencephalogram or EEG, which has got excellent temporal resolution, and functional MRI. Now, all of you are familiar with MRI scans. And if you think of an MRI scan as taking a picture of the brain, then a functional MRI are taking movies of the brain. And so we are using both the EEG and the fMRI to come up with signatures or high-dimensional patterns which can predict what we are trying to target. So, for example, we would set up a model, a statistical model, and say, can we predict what are the brain waves that are associated with moving quickly, or what is the fMRI pattern associated with moving quickly? Okay, next slide, please. So once we have these signatures in the EEG and fMRI of, say, moving quickly, then what we do is we give a subset, a tractable number of calibration stimuli. And then we set up a different statistical model to say what what is the effects on our previously derived biomarkers or signatures in the EEG and fMRI of these standard stimuli. Next slide, please. And then if we can combine these together, what we can do then is have the computer go through and generate millions and millions of potential stimuli and use that to predict what would be the result on our EEG and fMRI responses, and then what would be the result in the reaction time or whatever we're trying to optimize, such as moving fast. And this will, we believe, enable us to come up with an optimum or customized stimulus for that individual. Once we have done that, then we can transport or offload that optimum stimulus to a portable stimulator, which is step 4 you see on the slide. And then step 5, what we want to do is then test the hypothesis that these customized stimuli will be substantially better than the standard stochastic stimulus that's normally used. So next slide, please. So once we've already built a prototype, these are fairly simple devices, but basically they are constant current stimuli, which means they're not really constant, but what we mean is relatively invariant to the impedance. And although with the current prototype we have fits on a belt, later, of course, when we optimize this, we can put it into a different form factor. So the ultimate idea would be someone wears a hat or something with this stimulator with the electrodes behind the ear and with it uploaded to their particular customized stimulus. So that's the sort of thing that we're envisioning. Okay, next slide, please. So the next thing that I would like to talk about is monitoring people with Parkinson's. So one of the things that's a bit counterintuitive is people's motor performance can actually fluctuate hour to hour. And people can go from actually having excessive movement if they have a little bit too much medicine to not enough movement if they don't have enough medicine. And that window, which is called the therapeutic window, can unfortunately be quite narrow for some people. So some people have a very difficult time where they can oscillate between having too little and too much medication. And when we see people in our clinic, we may see them as infrequently as every 6 months or every year. And from that short visit, we're trying to extrapolate how they're doing throughout the year. And obviously that's very difficult to do. So there's been a lot of interest in monitoring people with wearable sensors. The problem is that after a while, it's hard to convince people to wire up and put sensors on every day. And so compliance is quite low. So there's a lot of interest in using video monitoring. But of course, a lot of people have some privacy concerns. And so we've teamed up with an ethicist, and we've had various focus groups with the Parkinson's community about what sort of video monitoring they would feel comfortable with. We think we found a solution in that what we do is we developed what's called privacy compliant monitoring. And I'll tell you a little bit more about that in the next slide. But I just want to point out that in addition to longitudinal monitoring, automatically assessing people has also some benefits. And this varies from country to country. In my country, in Canada, we have many sparsely populated remote communities. And so we want to be able to remotely assess people. In other countries, they have too few Parkinson's neurologists. And so they don't have necessarily the expertise to be able to come up with an assessment of the motor performance. And so if you could automate this process, that would be helpful. And finally, if you had a new treatment and you wanted to monitor people, tracking the disease automatically would be very helpful. Next slide, please. So here's an example of what we call privacy compliant. We need the high resolution camera to be able to automatically recognize the correct person. But in real time, what we can do is we can convert that person into a stick figure. And this is the only thing that's saved. So if anyone were to hack into the camera, all they would see would be the stick figure. And I think a lot of people are much more comfortable with this. So as I said, their images are never saved and never transmitted externally. They are just in real time with so-called edge computing converted into these stick figures. And this still gives us enough information to monitor the disease, yet preserves the privacy of the individual. So this is something that we're working on. Okay, next slide, please. So here's an example. What we've done is we've used commercial sensors. There are many companies now that are marketing these sensors, and we're using this as sort of a label or a gold standard. And we're also doing the video. And ultimately, what we want to do is we're using the sensors to train a deep learning or machine learning algorithm so that we can use the video to ultimately replace the sensors. So we would get the same positional information that we would get from the sensors, but solely from the video. Next slide, please. So here's an example of this in real time. You can see it's not perfectly registered. We still a few bugs to iron out, but you can get the idea that we can digitally zoom automatically into someone's hands, and we can look at the key points and then analyze these key points for mobility issues. And so this is the type of thing that we're doing. Next slide, please. And then finally, we're also looking at facial expressions. So people with Parkinson's have what's called a masked face. And so they have difficulty recognizing facial expressions, and also they have reduced facial expressions themselves. And what's kind of a fascinating aspect of the neuroscience of this is those two may be related, that in order to recognize someone's facial expression, you yourself have to have the ability to create certain facial expressions. And so we know that this is a problem in Parkinson's and in other diseases like Alzheimer's, people who may have difficulty with language. And so they may not be able to articulate that they are feeling anxious or unwell. And so we think some of these technologies will be helpful with facial assessment, especially if we have high resolution cameras that can automatically digitally zoom in and perhaps send an alert to a family member saying, grandma isn't feeling well or something like that. So these are the types of technologies that we're looking at. Okay, next slide, please. So that's all I had. I'm very happy to answer any questions. There's my email. So if anyone would like to send me an email later, I'd be happy to talk with them. So thank you very much. And I don't know, Manuela, how we moderate questions. Oh, there she is. Okay, great.
There I am. Thank you, Martin. That was really fascinating. So I think we have a little bit of a shy audience at the moment because I don't see questions coming through. So I'll profit to satisfy some of my own curiosities in the meantime. So I heard a story that your work on brain imaging was presented at CIA headquarters once. Can you tell us a little bit about that, or is it classified? Or
Yeah, so this is I think a bit of a funny story, but I was in San Diego working with a neural net pioneer, Terry Sinofsky, at the Salk Institute for Biological Studies, and we were looking at brain imaging technologies and brain imaging analysis methods. And when fMRI first came out, what you had to do is you had to hypothesize what the waveform would look like, and then search around in the brain for those parts of the brain that had that hypothesized waveform. So for example, if you had a task where someone was tapping their fingers and then resting, you would say, find me the voxels where there's activity and then rest and activity and rest. Well, the problem is what happens if you didn't know a priori what that waveform should be? In other words, if you didn't have a hypothesis-driven approach, how would you do a data-driven approach? And this is when we were the first to apply a technique called independent component analysis, or ICA, to fMRI. And this was a very nice data-driven approach to analyzing fMRI, which is now adopted throughout the world. What was interesting, though, is that the CIA contacted my supervisor and said, do you think this could be used as a fancy lie detector? So, of course, I'm a Canadian citizen, so they wouldn't ask me. So they asked my boss to fly up to CIA and present this new method for analyzing brain activity to see whether or not this could be a fancy lie detector. So that's the story of my slides being shown at CIA headquarters. I have no idea whether or not they used it or whether or not it was useful. I don't think it would be very useful as a lie detector, but...
Very curious to hear about it. Meantime, if we have a couple of questions on the vestibular waveforms and how often does a patient need the stimuli, every day, week, month, or are you still studying that part?
Well, that's a very good question. So it's interesting when you look at brain stimulus or stimulation. There's some stimuli that you give, and then you hope that there's a carryover effect. So, for example, transcranial magnetic stimulation, they use a very powerful magnet, but it's a huge machine, and because there's huge capacitor banks to generate the electric field or the magnetic field. So what you have to do is you have to stimulate people in the morning and hope that there's enough carryover effect to last for the rest of the day. We haven't gone that route. We've actually gone with something that would be potentially portable. So this would be something that you would stimulate them all the time. We think that effect is going to be much more stronger. There might be second order effects, but we haven't looked at that yet. We've mostly been focusing on the direct stimulation effect. So.
But I guess this is related to another couple of questions. One is that whether the patient needs to wear the device all the time, and a question that is about the cost of the technology, because it sounds amazing, but also very expensive, and especially the part about the customized brain stimulation.
Mm-hmm.
So maybe a comment on the outlook of it and which parts and which progress would help in making it also an affordable technology.
Yeah, well, I think it's premature about knowing the cost. Actually, the stimulators, of course, are very inexpensive. They're battery powered. You're stimulating a milliamp of current. I mean, it's not a big deal. The stimulation, you're right. We are using expensive technologies to customize the stimuli. But we're not at the stage. What we don't know is whether or not we could have maybe a repertoire of 100 stimuli, and all you would have to choose is which of the 100 is best for that person. We don't know that yet. So we're very much at the research stage where we want to find the absolute best stimuli, and that might give us an upper bound of what we can expect from the technology. And then what we may find is if that's too expensive, as I said, maybe have 100 stimuli and say, okay, choose which of the 100 is best for that person, and that may be 80% is good. We just don't know yet, but that's I can see that might be the way forward ultimately. So,
thank you, Martin. An interesting question is also whether the video monitoring could also help chart the progression of Parkinson's.
Yes, absolutely, and I think this is going to be. There's a lot of things that are. Where I think the benefit of this type of technology will be to find with data mining unexpected associations. So, for example, in Alzheimer's, if someone gets agitated, they may pace around the room more. And that's not something that you would be taught in medical school. So I think, it's finding these unexpected associations with certain movement patterns that might predict some aspects of the disease. Or, I mean, one of the challenges of Parkinson's is that it's a very heterogeneous disorder. And so it might be a way to stratify people and might be a way to predict whether or not some subgroup of people will respond to one therapy and not another. I think we're just starting to get into this, but I think we just need lots and lots of data to start doing the data mining. And I think some of these associations will start to fall out.
There's a question from the public which actually sort of ties with a question that I had myself. So the question is, how come the people who are diagnosed with this type of disease can remember very old times and not recent time? How this is really related with the chemical reaction in the brain? You mentioned at some point also Alzheimer when we were talking about facial expressions expressing feelings, but I guess also, for example, multiple sclerosis or other diseases. I mean, there's similarities across all these, so I guess integrated research could help and be beneficial to more than just one disease.
Yeah, well, I mean, we in clinical medicine, we often talk about whether or not people are lumpers or splitters. In other words, whether or not you try and look for commonalities across many diseases, or you try and find things that discriminate between the different diseases. And it is true that there are certain commonalities across the diseases, but there are also some very specific things. And I think your question was about dementia. Is that right? About recalling things remotely, but not. Yeah, that's very fascinating. And basically what happens is that there's a part of the brain that encodes new memories. And so there's a whole area of what's called memory consolidation, where something happens, it's in a transient buffer, and then it basically gets translated into something that's much more permanent. And if you damage the area that's encoding the new memories, then, which is what happens in Alzheimer's and other types of dementia, then you have the consolidated memories because they were previously consolidated, but you lack the ability to encode new memories. And so that's why it's recent memory that's quite characteristically affected in Alzheimer's and similar diseases.
It's fascinating. I mean, just like when you mentioned that there seems to be a connection between the ability to have facial expressions and the ability to recognize the facial expressions. So, I mean, these are areas in the functioning of the brain that are really, really fascinating. I had the questions because you mentioned several times, of course, functional MRI as well. Are you happy from the point of view of your research with the resolution of the current MRI machines? Is there something that you would wish to have to guide you better in the understanding of the brain and the diseases? It's a physicist question, of course.
Yes. Well, of course, more resolution is better, but the problem is with fMRI, you're actually measuring changes in the blood deoxygenated hemoglobin. So you're actually indirectly measuring blood flow. So the resolution may actually be on the physiological basis of that, not necessarily the physics of the machine. So I don't know if necessarily much stronger or much greater spatial resolution would necessarily help us. I think structurally it's very helpful.
Thank you, Martin. Thanks for answering the curiosities of the audience and also some of mine. And I guess it's time to move on to the next talk. So it's back to Anelia for the continuation of the session.
Okay, my pleasure.
And thanks to our speaker. Thank you all also very much for understanding and your patience. Kitty Lau, the former founder and the CEO of Ideabatic, will actually give her talk at 4 p.m. this afternoon here in the Globe. In the meantime, we're continuing with the medtech sector, and to moderate this next talk, I'm delighted to introduce Roxana Rata. Roxana is a radiation protection physicist and with a real interest in accelerator applications in the medical field. She did her PhD in radiation protection for proton therapy, and after that, she joined CERN as a senior fellow. After CERN, Roxana worked as a radiation protection specialist for the British government. 1 year ago, she joined Varian Medical System as a beam physics engineer for the proton therapy systems there. Roxana, the floor is yours.
Thank you so much. I am delighted to be here today. Thank you so much, Anelia, for the introduction. And I guess that now it is my turn to introduce the new guest. His name is Markus Wager, and Markus has experience of more than 10 years in healthcare. He started with commissioning one of the treatment facility, and after that he joined MedAstrom team at CERN, where he developed a new software that generates the accelerator setup for the large beam in use for cancer treatments. So to understand more on radiation therapy, he continued his studies doing a master in radiation therapy. And now he's working at Cozy Lab, where he's helping other clients to develop innovative treatments. So Markus, the floor is yours.
Roxana, thank you for this nice introduction. Yeah, you mentioned some of the aspects that I'll talk about. Also the work at CERN, which I'll explain in a bit more detail. It is correct. I used to work on the accelerator side with the accelerator control system, on controlling really all the different elements of an accelerator to get them to work together and produce a decent beam. Often enough, that is not easy, right? We had days where we were really struggling to get a nice beam out of the accelerator. And so I kept asking myself, how is this beam then used, right? Proton particle therapy is one of the most advanced therapy forms for cancer that is available today. So how does that all work? And yeah, so I decided to go back to university to find out more about this. And yeah, for today, I thought when I worked at CERN, I was really curious about how beams are used for medical treatments. Today, I know more about it, so I'll share this knowledge, hoping that you find it interesting as well. And so in my talk today, I will take you from the world of accelerators into the world of cancer treatments. That is the idea. But let me start with the accelerator side. And on this slide here, I have a picture of the MedAustron accelerator. So MedAustron is a treatment facility in Austria. They are treating patients with protons and since some time also with carbon ions. And the MedAustron accelerator was built in cooperation with CERN. So some years ago already, CERN developed a study investigating, how synchrotrons could be used medically. And then MedAustron, when the decision for this accelerator was taken, they agreed with CERN to work together to collaborate on building the actual synchrotron. And so a large number of MedAustron employees spent time at CERN. I, in particular, worked in the accelerator controls group. So I worked with Johannes Gutleber from CERN, some of you might know. And so the MedAustron machine is a synchrotron machine that we built, and I mentioned it can produce proton and carbon beams. Those are the most typical beams that are used for cancer therapy today. If you look at the machine with the CERN knowledge in the back, it's maybe not that interesting a machine, right? It's 78 meters circumference. It can produce protons with up to 800 MeV. So compared to CERN accelerator, not that much. For clinical use, even these 800 MeVs are way too much. 250 MeVs are enough. So from that perspective, perhaps medical synchrotron is not that interesting. But there are some factors that make it a challenge to use synchrotrons medically, and I'll explain how come. The first thing that I need to explain is how the beam of the synchrotron is actually used in a medical treatment. And I have this on the slide here. So the beam from the accelerator is this green line in the picture. It's delivered to the patient and to the tumor volume. We call it typically a pencil beam. It has a size of about 5 millimeters typically. And now particles have a very special, very distinct way of interacting with matter. So particles that travel through tissue, they will deliver some energy, they will slow down gradually, and then shortly before they come to a stop, they deliver all of the remaining energy in a very, very short distance. And this distance, distance is here in the diagram. It's this peak. The peak is called the Bragg peak, and this peak is what makes particles interesting for treatments. The peaks allows to focus the energy delivery of the beams to a specific region. The area to where the energy is delivered, this peak depends on the initial energy of the beam. So the higher the energy of the particles, the deeper in tissue or in matter really this peak will be produced. And in a treatment, the energies of the beams are chosen such that they put this peak into the tumor. And that then means that the dose is delivered to the tumor, but after the tumor, there's really not much energy, or in fact, no energy at all that is delivered. And that, of course, helps to focus the energy of the tumor, of the energy of the beam to the tumor, and spare the tissue that surrounds the tumor. Right. That brings me back to deliver energy to a bigger region, to the target region. It's not enough to use only one beam with one energy, but beams with multiple energies need to be used to stack the beam. Right, the Bragg peaks are thin. The pencil beams are not that thick, so to cover a bigger area, the beams are scanned, different beam energies are used. And that, in fact, is one of the challenges of a medical synchrotron. So the synchrotron does not only need to be set up to provide one energy, it needs to be set up to provide many energies. For the clinical use, I have the examples here, 60 to 200 MeV. The spacing between two slices is perhaps half of MeV or 1 MeV. And so now all of the elements of the accelerator, they need to know what to do to deliver a beam of a certain energy. So the Med-Austron accelerator has several hundred magnets, different RF structures. The timing needs to be right. And all of this needs to be calculated for the different particle types, for the different energies, the different beam sizes, even the intensity of the extraction. And so what I did at CERN, I worked with our accelerator physicists, and I developed the software that does these calculations. So in that software, the physicists would enter, for example, the tune or the chromaticity of the synchrotron, and the software would then in the back use MATX, would do some optics calculations, and would produce the waveforms and the set points for the different magnets and elements of the accelerator, would produce the timing sequences. So this worked very well. Here are the results. It's really a measurement of the beams at Med-Austron. Med-Austron started treating patients in 2016. So the software that I developed, the cycle configurator, obviously worked nicely. It helped to set up the accelerator to treat patients. I think by today, Med-Austron treated several hundred patients. And it's, of course, not only a success story for the work I did, but I think a success story for CERN in general, right? For the knowledge transfer of CERN and the positive impact that CERN and studies like the PIMS study that investigated the use of medical accelerators and that was produced at CERN, that they really have a positive impact on society. Now, what I promised you is that I take you from the accelerator side to the medical side. And to do that, I first need to explain how particle beams produce some treatment effect, right, a medical effect. And the starting point of that are ionizing events. So what happens is that particles interact with electrons mostly. For example, they can kick an electron out of its place, causing an atom to become an ion. But electrons are used in the bonds between atoms, in the bonds between molecules, and molecules forming important structures in the body. The DNA of a cell, for example, which I have here on the figure. And the energy transfer of the beam now causing these ionizing events can mean that these bonds are messed up, that the bonds between important structures in the body are broken, which can mean, in case of the DNA, that there is damage caused to the DNA. Now, cells can repair DNA. Of course, this is important, otherwise we wouldn't live. But if enough of this damage is caused, then cells cannot repair it anymore and they die. And if it's the tumor cells that are damaged and die, then we have the tumor treated. Of course, this is not the entire story. Yeah, the ionizing events are a starting point, but the way that cells really react to these events, they depend on a lot of different factors. They, for example, depend on the particle type. They depend on the type of the cell, right, on the type of the tissue. They even depend on the amount of oxygen that is available in cells. And then also cells don't live alone. Cells live in big structures. They form organs, right? They communicate with each other. And they also communicate with the immune system, meaning that also the immune system starts to interfere with the reaction of the body on the radiation that is delivered. The reaction of the immune system is something that we only starting today, I think, to really understand. And all of this together in the end, all these factors together in the end, then cause a biological response, which means, well, that the tumor dies and that the patient can live without the tumor. In the medical community, this is called a disease-free survival. But the lifespan, of course, is not the only aspect. It's also a question of how good is the life, how much side effects does a patient have. And this knowledge really of how radiation affects the body as such and the biological response it causes, that is now knowledge that oncologists have, right? That is the knowledge of medical doctors. And for a treatment, the medical doctors bring their knowledge in, they diagnose the patient, and they define the type of treatment, they define the particle type to be used, they define the doses to be delivered, the amount of radiation. And they would also define the target volume, of course, right? The part of the body that needs to be treated. In my example here on the slide, this is a CT scan, and it shows the prostate in the middle in red, which in my examples will be the target volume, that it's a prostate treatment. So oncologists would use a special software treatment planning system and would in this software actually draw the volumes that are interesting. They would draw the target volume, but they would also draw structures that should not be irradiated. The rectum, for example, which is the brown part, is down here. The rectum is very sensitive to radiation. If the rectum was irradiated, it might give a patient severe side effects in going to the toilet, problems with that. And that is, of course, something that we don't want to have in as an outcome of a treatment. And so what happens in treatment planning is that these aspects are considered, that oncologists prescribe the targets and also mark the volumes, that targets that should not get those. And then the right setup of the treatment is determined. I have two examples here. I have one where it's 4 beams coming, 2 from the sides, 1 from the front, 1 from the back, compared to one that only has 2 lateral beams here in the center. And so the question, in both options are feasible, really. The question is, which one is the more suited for a treatment? That's what's determined in treatment planning. And I can tell you the more suited is the case with the 2 lateral beams. The reason being that, again, we want to avoid those being delivered to the rectum. So a beam that comes from the back, for example, would exactly deliver those to that structure and should be avoided. Whereas from the sides, the beams go through the bones here. This is the femoral head. And the bones in adults are not very sensitive to radiation. They can take a lot of radiation without side effects that occur. So the better setup in this case is with 2 beams from the side. That is what is defined in the treatment planning system. The treatment planning system then calculates the dose, and it calculates which energies and which beams it needs from the accelerator. And that brings me back to the accelerator side. So I have here the accelerator of a facility in Germany, Heidelberg, Ion Beam Therapy Center. And what I think this slide really nicely shows are the dimensions of this treatment, really. The Synchrotron being a complex, bigger machine compared to the patient. The beam is accelerated, is then delivered to the patient, as I've shown on previous slides. But then the particles, when they leave the accelerator, they go their path, right? They basically go straight. And so what we still need to do is we need to get the patient to where the beam is, get the tumor really into the path of the beam. And this is the last part that I want to show you in my talk now, how the patients are set up. So patients are positioned on a robotic system. Yes, a robotic arm. Again, these are examples from Med Austron. Patients are positioned on the treatment couch. Typically, there are these, for head and neck treatments, typically these face masks are used. So they are a plastic mesh that is heated in warm water. When it's warm, it becomes soft and it can then be pulled down over the patient's face. It's mounted in a certain position on the treatment couch. And then it gets cold and it remains in this form. And then every time the patient comes for the radiation, this mask is placed over their head and mounted on the table again. And that ensures that the patient is really lying in the correct position on the couch. There are some other options. You could use a vacuum mattress. So it's a foam mattress, and air is sucked out. And then it keeps the form of the patient. It's another device. Or down here is a knee rest, also a device that can help to position the patient in the right position for the treatment. You might still ask me, Marcus, this all looks like it's not very accurate. How can you, accurately treat the patient? And I'll show you the answer to that. In fact, all aspects of a treatment have uncertainties. That's important to know. It starts with the prescription of the oncologist, right? Different oncologists would draw tumor volumes that are a bit different. It also includes physical aspects, like the proton range, for example. It's not clear where in the body protons will stop. There's certain inaccuracy. And then it's the setting up the patient on the robotic couch, immobilizing the patients. All of this has some uncertainties that need to be addressed. And well, the way to do that really is to consider it in treatment planning already. And in fact, the way out is to irradiate a structure that is bigger than the actual target volume. So in the treatment planning, bigger volumes are designed that include additional margins to account for those uncertainties. So in the end, we'll deliver a bit more radiation to a bigger volume than strictly necessary, but that helps us to ensure that we really cover the tumor volume with the required dose, even if there are some uncertainties in the treatment. So with this, I think we started talking about the accelerator setup, the different energies. I explained a bit about the delivery of the beam. We looked at how patients can be set up. I explained the medical aspects with ionizing events and the biological reaction, how this is used in treatment planning. And so in the end, the setup of the accelerator determines the biological response. And we looked at this entire chain of how particle beams are really used to treat cancer. It was only a short overview. There's, of course, a lot more we could talk about. I hope you still found it interesting. You have some opportunity to ask me more. I will be in this networking session at 4:30. You can also find me on LinkedIn or on the alumni portal. I put also the link here of this master program that I did. It's a program taught in English. It's in Austria, but it's taught in English. It's a part-time study. So if you're interested in radiation therapy and in learning more about it, perhaps this program is something that is of interest to you. Also, today I'm working at COSILab, which is a really interesting company. We are very much involved in particle therapy and in new ways of using it, very innovative solutions. There's a lot of open positions. If you have any interest in the software domain and working at COSILab, check out those positions. If you'd be interested in working with COSILab on some particle therapy related field, I'd also be happy to hear from you. You can contact me at my COSILab address, which I put on the slides here as well. And that is really the end of my talk. Thank you for listening.
Oh, thank you so much, Marcus, for this very well done presentation, very precise and very good information on it. So we have now 10 minutes for questions. So feel free to write your questions on the chat if you have one. Yeah, meanwhile, I have some questions for you, Marcus, if that's all right with you.
Sure, great, Roxana.
So I'm not aware of Astron, but do they have a research room?
They do have a research
That's
room as
why,
well, yeah.
yeah, okay, and
That's
that's
why I
the
mentioned
maximum
these.
energy used.
That's the maximum energy
Okay,
used. Those are these 800 MeVs. Yeah, for a treatment, it's way too much energy. It would put these Bragg peaks outside of the body, but that's not what one is interested in, right? We want to have the Bragg peak in the body, in the tumor volume, really. And for that 250 MeV, they can deliver beams already to some 25, 28 centimeters depth in water, and that is really enough for treatments.
yeah. Do they use carbons as well, or
They do
or
since I think 2019. They also have carbon ions set up, and they are using them for treatments.
okay? I was not aware of that, so it's something that came 2 years ago, let's say.
Yeah.
Okay. In your opinion, what are the things that could be improved in particle therapy?
Yeah, there are some aspects. The one is that it's still a bit more expensive than other forms of therapy. So one big goal would be to make the treatments cheaper, which would mean that the accelerators, for example, should become cheaper, smaller, simpler than this complex synchrotrons used at Medaustron. That is one aspect. Another aspect is that this biological response is still not understood too well. So there is a lot of research going on in that area, the reaction of the cells and the reaction of the entire body and the immune system to radiation. There's a lot of research going on in that area, and some new findings coming out, examples, flash therapy or microbeams that can, with even less irradiation, reach very good treatment outcomes. So it's ways to spare even more healthy structure to deliver less dose and still reach even better treatment outcomes. These are things that are investigated at the moment, and that I think will be important in the future of particle therapy.
Yeah, I see. What things do you think that scare people most when they are having treatments with protons, for example? I mean, the side effects. Uh, in your opinion, the side effects are something to be scared of? Because many people, they are asking me, for example, oh, if I am irradiated, what will happen after? So do you think that people should be scared about the side effects after proton therapy?
No, I think there is no reason to. This is the main advantage of proton therapy, that the side effects are very, very limited. Very typical side effect would be that the skin gets rash, gets infected, because there's always some energy delivered superficially as well. But these are minor side effects. Otherwise, I think exactly proton therapy has a very good sparing of healthy tissue. In treatment planning, they really make sure that the critical structures are spared. Right, I showed this example of the rectum. So they check some dose will be delivered to the rectum as well. That can't be avoided, also because of the margins that are necessary to account for the uncertainties. But in treatment planning, they ensure that this is only so much dose that the rectum can tolerate, and then it's overall over the entire treatment. It's not. It won't have bigger consequences for the patients.
Okay, thank you so much for your explanation. It seems that we don't have questions.
If you have a few more minutes, there
So
are some interesting aspects going on. Also, CERN is still very actively involved in this community. I can mention that. So they did this PIMS study for this accelerator that the Med-Austron accelerator is based on. Today they have a study called NIMS, New Ion Medical Machine Study. That study is in particular investigating the production of carbon ion beams and what are the best accelerator techniques to do that for treatments. Other examples are this flash therapy that I mentioned. CERN is involved in that with, I think, a university in France. So flash therapy delivers very high doses in a very short time. Something that also CERN is involved in with the accelerator technologies. There's also some spin-offs, like companies like AVO, Adam, that are working on producing accelerators that are smaller. They're using a LINAC that is very small in its footprint. And it's a spin-off of CERN, so it's also technology developed together with CERN. So CERN is still very active in this field, and I think there's a lot going on in terms of particle therapy today. Still, it's a very interesting field, really.
we have some questions in the chat. So the first one is: it is necessary to build a new building or even a new hospital to place the accelerator?
Yeah, not necessarily. A big machine like the Synchrotron from Med-Austron needs an entire building. But what I just mentioned, AVO, for example, with this LINAC, they are planning to install this LINAC in existing buildings. And it's not only AVO, there's other companies, Proton, for example, as well, that build very small accelerators that can be installed in existing treatment facilities, even in existing treatment rooms.
I see. The next question is: which is the average hydrotherapy treatment duration?
I'd say something like 20 fractions. The radiation is fractionated, so the patient has to come 20 times. Usually this is done on the work days, so 5 times a week over 4 weeks. That's a typical course for a treatment.
I have another interesting question. Is an online monitoring of the deposited dose feasible? Could it help reduce the side effects by adjusting the near real time the patient position?
Interesting question. Yeah, there are some ideas to do that. It's not trivial, but there are, especially with carbon ion beams, there are some PET nucleate PET radiation that is produced and that could be measured in real time. There are other ideas to measure the dose. There's, for example, proton imaging, where the proton beam is really used not only for treatment but for imaging as well, and that could really help to figure out where in the body the dose is delivered. There's a lot of research going on in this area, really, to find good solutions.
I see. Yeah, this is all. Yeah. Thank you so much, Marcus. Thank you so much for your presentation.
Goodbye.
Bye.
Thank you, Roxana and Marcus. You can now continue with your questions in the networking tables at 4:30, where our speakers will be available for more questions. In the meantime, we have come to our final talk on this topic, and to moderate this next talk, I'm delighted to introduce Maurizio Vretena, an accelerator physicist who joined CERN in 1988 to take part in the design, construction, and operation of linear accelerators. In the recent years, he has been increasingly active in the field of societal application of accelerators, particularly in the medical field, where he is currently leading the Next Ion Medical Machine Study, a new CERN initiative in the field of accelerators for cancer therapy with ions. Thank you, Mauricio.
Thank you, Anelia, and we are really excited about the next talk that is from Kitty Liao. Kitty Liao is the founder and CEO of Ideabatic. That is a new company that has already been awarded many times for its work. Something happens? I don't know. Okay, so you move forward or I continue? Yes, yes. Okay. Sorry, because as we have noticed, we had a number of technical problems today, and now we are trying to reach Kitty, who is not ready to start. Anyhow, I have the pleasure to introduce her because this company, Ideabatic, is really very interesting because Kitty is bringing to the company the competence that she has developed in superconductivity on the accelerator field, in particular developing diagnostic systems as a fellow at CERN for many years, taking this to the vaccine cold chain, which is, as you know, a very important topic in this particular moment. And her company also has a social side that is also very interesting. So I hope that we can get in touch with her soon so that we can start. But in the meantime, I can also say that what I really like is that the motto of her company, Ideabatic, is innovative humanitarian engineering. And so the combination of these three aspects is something that makes really it's very interesting. So a social enterprise of high technology. So is Kitty online? No. Okay. Yes. Yes. Because yes. Because we are supposed to start now at 4. We had a new timing. All right. So let's see. Can wait a few minutes more because it's always complicated to reschedule a talk online like this one. So we have now a few minutes. Yes. No news, I think, for the moment. It's 4 o'clock. So, okay. Still waiting. Maybe I can reconnect to what the previous speaker said because he mentioned an initiative at CERN, Next Time Medical Machine Study, that I have the honor to lead. So simply, I can just stress that we try at CERN to work on the critical issues for ion therapy, in particular for carbon therapy that were mentioned in the talk. So there is an effort at CERN. So we try to do in the limits of our limited resources for medical applications. But this is an important activity for CERN, and I think an important initiative in the field of trying to design an ion therapy system of next generation. So go beyond the old PIMS study that, as some of you might know, was developed at CERN in the time between around the year 2000, let's say. So it's 20 years since these machines have been designed, and we try to work in the direction of machines of applying new technologies from particle physics, from the new generation of accelerators to these machines. So Kitty is not yet there. No. Yes. Yes. Let's wait a couple of more minutes. Yes. Okay, great. So, Kitty, so it's a pleasure to have you here. And when you're ready, the floor is yours. Okay.
Thank you. Can you hear me? Thank you so much, Maurizio, for the kind introduction. I cannot see my slide at the moment, so there it is. Great. So good afternoon, everybody. My name is Kitty Liao, and I'm the founder of Ideabatic. Today, I'd like to share with you my work creating solutions to an important global problem. And firstly, I suppose it's a good idea to start with my connection with CERN. So as Mauricio said, I was a CERN fellow in 2010 to 2013 in the Beams department working on superconducting cavity diagnostics. So the picture on the works. The picture on the left kind of sums up the systems I developed. A second sound detection system that detects a quantum wave called the second sound to triangulate where the quench locations are on these superconducting cavities. And then the second one is an octopus temperature mapping system that I created that has the capability to test out the temperature hotspots on different cavities with different shapes and sizes. And so all of these systems are used in superfluid liquid helium at 2 Kelvin. And so picture on the right, that was a farewell party at CERN back then. So yeah, that was a long time ago, but I really enjoyed my time at CERN. And then so I moved on to do more kind of low temperature research in the US. And 7 years ago, so in 2014, I participated in a humanitarian hackathon organized by the Port organization. So the Port was founded by CERN people back then, and they sat down with NGOs like the WHO, Red Cross, and Gavi, these kind of people, to look at the most urgent problems that needed solving. And they defined the challenges. The Port, they bring people together from different backgrounds to work on these very important problems during the hackathon. And so at the time, I was really shocked by the information I found out. So in 2014, according to the WHO, 18.4 million children died. 18.4 million children had no access to basic vaccinations. And each year, between 2 and 3 million died of a vaccine-preventable disease. Before COVID-19, the WHO reported that globally, 20 to 50% of the vaccines were spoiled. But in reality, the spoilage is much worse. The data in remote areas is very limited, with one particular remote area reporting an 86% of spoilage rate. Oops, sorry, back to that slide. So using just 20% of the wastage in the calculations, this equates to at least $1 billion wasted every year. And our research in remote Nigeria and Madagascar revealed spoilage rates of 100% in some instances. Today, excluding COVID-19, the number of unvaccinated children has gone up to 19.9 million. And a major reason for this is that during the last few miles of delivery, the so-called last mile, vaccines become damaged by temperature fluctuations. In other words, they either become too hot or too cold. With my background in cryogenic systems and in innovation, I thought there must be a way to solve this. This is why I founded Ideabatic, to solve important problems with smart innovation. And if some of you are also from engineering background, you probably guess where the Ideabatic name comes from adiabatic. So what is a typical last mile journey? Trained health workers, they carry cool boxes with ice packs in extreme weather and over difficult terrain, by foot, bike, motorbike, donkey, canoe, or a combination of these, as you can see from the pictures. Bear in mind that vaccines need to be kept between 2 and 8 degrees before injection, and these journeys can take up to 6 days. And however, existing solutions either freeze vaccines or last barely a day when in use. this slide shouldn't be here, but let's skip that for now. So these are the commonly used vaccine carriers. Again, these are essentially cool boxes filled with ice packs, as you can see from the pictures on the left and on the right. They share the same profile in the middle of the screen. The majority of these cool boxes, they freeze vaccines at the very start, as you could see, the first black circle. And all of them suffer from overheating earlier than expected. So the claimed cooling duration from these manufacturers is from 17 hours to 50 hours. But this is much less in reality. Why is that? Oh, 1 second. I'm just checking my clicker. Sorry, my phone is a bit weird. Yes, there we go. So one of the biggest issues we observed in last mile vaccination is human error. This is what we captured in Nigeria. The trained health workers often just left the lid completely open throughout the injection session. You can see here these cool boxes. They require 4 ice packs, as you can see in those slots, the blue areas. They're designed to accommodate 4 ice packs, but in the field, it's common that not enough ice packs were used. So I'm just going to show you the clip very quickly. So note that the health worker was only starting to write out something on the vaccination records, but he already opened the lid and just waiting there. So you could see, and it was 36 degrees outside. So what happens in these cool boxes in the field in terms of their performance? We took 2 of these models back to the lab and performed experiments and analyzed them. You can see that using 1 ice pack means that the cool box goes down to 17 degrees only for a few hours. Using 2 or 3 can keep things cold briefly for a few hours, and really you need 4, which is the required number. But then the vaccines are exposed to freezing risks at the very start, and this will spoil certain vaccines. In the developed world, or in areas with better practice, health workers, they would condition the ice pack first, which means they take out the ice pack from the freezer and wait for a couple of hours before putting the ice pack back in to avoid that freezing issues. So now, let's assume that all health workers place the correct number of ice packs in the cool boxes, and they open the lid and close it every time when they use it. What do you think would happen? So the curve in red, this represents the humidity. So the humidity goes up and down when you open and close the lid, as you would expect. And for the smaller box, which is the dark blue curve, within an hour the temperature had already exceeded that maximum temperature when they do the opening and closing behavior. And then the bigger one, the grey box at the bottom, that's within 4 hours. So in reality, these vaccine cool boxes are not able to keep vaccines cold for long enough after the start of use. Not to mention, often the door was left wide open throughout the session, as you saw, and other errors that can lead to risks of spoilage. So what if we could reduce the wastage to less than 5% and extend that journeys to 6 days? And most importantly, we know that a functioning device can still fail when it's not used properly. So even when we ensure that our device doesn't spoil vaccines, can we ensure that it will still work even when it's not used properly? Our solution is SMILE, a smart last mile cooling system. SMILE is designed to be fail-proof, and it keeps vaccines at the right temperature for 3 to 6 days without external power. So today I pick out some of the features of SMILE with a side-by-side comparison, so you could have a better understanding of how we design these things. So first of all, leaving the lid completely open throughout the session shortens that cooling duration. And Smile has a self-closing door. So that means it's okay to forget to close the lid, and we can minimize that heat transfer. The second one, using an incorrect number of ice packs can lead to spoilage. So the Smile single ice pack design means it's impossible to place too little or too much ice. It's the same procedure, putting the ice pack in the freezer, taking it out, putting the ice pack in. And there's no hesitation putting 1 or 2 or 3 ice packs. The other advantage is that Smile is freeze-free. So the users, they don't have to condition the ice pack, no need to wait for the ice to melt before putting it in. So once the Smile ice pack is out of the freezer, it can be used straight away. It saves time and reduces error. So the last one on this page, some vaccines, as you can see, they're in direct contact with the ice pack. So this can lead to the freezing issues. Our ice pack chamber is separate from the vaccine chambers, as you can see, so there are no freezing issues. Next one. Right. So in the current cool boxes, glass vials and ampoules are tightly packed, and it leads to breakage issues. And in our design, all of the glass vials and ampoules are held securely and separately by our sample holders to prevent breakage like these in these cool boxes. So I'm just concerned with the time. Is this timer correct that I've only got 2 minutes left? So if that's the case, I'm gonna not show you some of the prototypes we developed, maybe when we have some more time later. The second one on this page, repeated opening shortens that cold life. And for Smile, with the self-closing door and an air-locking carousel mechanism, that means we can ensure that the temperature is still maintained even during use. So you're only exposed to that one chamber at a time, and then the rest stay cool. So users, they can look at the vaccine record on the backpack flip on the left of that picture here. So for example, they put polio vaccines in chamber 1 to 5, and some vaccines in chamber 6 to 9, or COVID vaccines in 10 to 15, let's say. So they can just turn the dial to the desired chamber and open the lid, and then go on with vaccination, and the door would close itself. So currently, excuse me, currently these vaccine cool boxes have either crossbody or top handles, so it's really awkward to carry on some of these last mile journeys. Also, it requires several people on the trip, as you can see here, one carrying the cool box and the sharp disposal box, one carrying syringes and dry goods, and one carrying vaccination records. And Smile has an all-in-one backpack, which is comfortable like your mountain backpack has a hip and chest belts. And it's hands-free for walking in mountains and crossing rivers. As you can see, the health workers carrying Smile in the field. And one Smile can carry over 500 doses, which is more than enough for these last-mile journeys. It's also suitable for large-scale vaccination campaigns. So Smile's help is not just limited to remote areas or developing countries. Did you know that biomedical scientists and researchers all over the world still use cool boxes to preserve their samples? Yes, even in developed countries like the UK and Switzerland. These cool boxes often hinder important work due to their unreliability. So if you develop temperature-sensitive products and would like to work with us, then let's have a chat. So this is our journey. The Smile has been iterated, lab tested, and field proven. As part of the Expo 2020 Global Innovators, we are invited to exhibit there in Dubai in January 2022, and we would love to see you there. So finally, thank you very much for listening to the session today, and let's spread Smile across the world. Thank you very much.
Thank you very much, Kitty, for your talk and for your smile. I think very simple and very effective. So I'm really impressed. Great. So we have already some questions. No, we don't have questions in the. So don't see questions in the sheet. But maybe I have a question for you. I think something that was. I mean, I also like your procedure on how the different steps that you followed to come to this SMILE product. But I think very important was the analysis on the field. I mean, how much time did you spend on the field just looking at how the vaccines were handled in some countries? And maybe additional question, who is funding that? Was you were already doing that with funding a company in your mind, or was a kind of a first approach? Okay, so if you can develop on
I
that.
don't quite understand your second question. Could you clarify, or should I answer your first one?
Answer the first one, and then maybe we can go to my second, please.
Yeah, sure. So at the start, when I started working on this project, it was a lot to do with interviewing people on the ground. So really interviewing them, what's happening, and then seeing the footages. But of course, that is still not the same as going out there and seeing how things are. And so as I kind of progressed, and then winning some grants and prizes, and that really enabled me getting the team and people to go on to these trips in Nigeria and Madagascar to really see how things are. And so the time we spent in the field, I suppose in total I just many hours just really observing them. A lot of them is to just observe how they do things, and then to look at the behavior. And because it's just so different from where we are, and a lot of the things for us, it's we're trained to open the lid and we close it. That should be very easy to do, but things are just not happening there. And so just from really seeing how things are in the field, and that really give us a strong kind of verification on focusing on the human problems. How do we design a system that can help eliminate that kind of error, or at least reduce? So your second question, sorry, could you repeat that, please?
No, but you answered that already. I mean, who was financing this field analysis? And you said your first awards just gave you the opportunity of doing this field analysis. I think it's very important.
Yeah, I think funding is always very challenging when it comes to a hardware product like this, because when we want to prove our concept, we need to make it, and we need funding for that. So at the start, it's winning some small prizes, and yes, that does help with some of the prototyping. But then when it comes to really testing out whether it works or not for our product, we can't really use things like 3D printing, because 3D printing it's kind of based on the additive kind of technique. So if you use that, things are added, layered up like this, and they look like they're one piece, but on a microscopic level there are holes in them. So for something that needs to be thermally robust, we can't just use some 3D printed part. So all of our these thermally critical parts, we use machining. So a machine cuts out the shape from a big block of plastic, so it ensures it is one piece, and then it's the same as what it will be when it's mass produced. Funding was a critical thing, but luckily along the way we've been winning some grants and things, so that really helped kind of iterate and then test out things in the lab, in the field.
Okay, great. So some questions are popping up, but first of all, I read a comment. No question, just compliments for the super presentation.
Oh, thank you very much.
One of the comments. Yes. Then the question is, what are the biggest challenges that you faced while building this prototype? What was challenging?
That's a great question, because I prepared something I wanted to show. I think it's more technical, more interesting, and so yes.
And you have only 2 minutes to answer this question.
Oh, okay,
you have only 2
great.
minutes to answer this question, unfortunately.
No problem. So, as I was talking about in SMILE, we have sample holders to keep the vaccines in place, because at the moment they just shuffle all the vaccine in the box, and then things can break, so they have to work really carefully. So I'm going to show you like the first few prototypes we've got. So you see, this is like, so this is just a segment of it, and you could see that this just the vaccine can be in there, but you could see the material needs to be flexible enough, but it also has to be strong and rigid. It also needs to be durable. As you can see, the field, there's dust and dirt can come in, and then these things will not be working anymore. So all of that as we need to consider. So from the first few versions, this was like a belt structure. So that means, say, this chamber, if it's broken, then we need to replace the whole belt, which isn't ideal. And after some more few, this one is slightly better, more iterations, we have now come to a two-part structure. So you can see this, the black bit is the rigid part, and then there's a separate holder. So I'll just do that. So you can see there's like a fin kind of thing, and that can be fitted onto this. And then so one of the challenges that there are hundreds of different packages of vaccines. So they come in these small sizes like this. There's a big one like this. There's ampules like this, and there's big ampules, all kinds. So how do we make a universal one that all of these vaccines fit? And so that's another development challenge. So we've gone through various iterations of these holders, and then you could see they're kind of flexible. If I
the time is almost over. Sorry, but
so
yes.
So now
I'm afraid that
we have a holder that is universal for all kinds of things like this as well. This is biosample tubes, that's vaccines, and then we can put ampules in as well. So yeah, this one I would say is one of the most challenging one among the many.
Yes. Okay, so thank you very much also for the demonstration of how it really looks like. So really, it was a very exciting talk, and we wish you all the best and all success with your idea. We are all behind you. Just many thanks, and thanks again for this great presentation. Okay,
thank you
very much, Kitty.
everyone.
Thank you very much. Bye-bye.
Thank you. Bye.
Bye.
Thank you to all our speakers, such fascinating talks, really demonstrating the motto of Second Collision event, Research Matters. You can now join some of them at our networking tables, which will open in 5 minutes at 4:30 CET time in Restaurant 1. You can also visit our booths in Idea Square or visit our virtual experimental areas. Enjoy the platform. See you later.